A very warm welcome, everyone. I am Ellen Herman, co-director of the Wayne Morse Center for Law and Politics and professor of history here at the University of Oregon. We're so glad that all of you could join us. We hope that each of you and all of your loved ones are in good health. The mission of the Wayne Morse Center is to promote civic engagement, inclusive democracy, and justice by bringing together students, scholars, activists, policymakers, and members of communities, people just like you. We discuss a wide range of issues affecting Oregon, our nation, and the world. We work in the tradition of Oregon Senator Wayne Morse, who believed that political independence and education were building blocks of justice and democracy. This panel on the history and future of racial science and eugenics is part of the center's Science, Policy, and the Public theme of inquiry. If you aren't already on our mailing list and would like to hear about other future events like this one, please sign up for the mailing list on our website. Just go to the website and you can type in your email address at the bottom of the home page. I'd like to thank our co-sponsors for this event, the University of Oregon Department of Indigenous, Race, and Ethnic Studies, and the Black Studies Program and Minor. We are also very grateful for support through the Lorwin Lectureship on Civil Rights and Civil Liberties. A thank you, as always, to the amazing members of the Wayne Morse Center team, Thea Croman, Rebecca Dinwoody-Flynn, Abby Tilley, Dan Titchener, and Christine Waite. Literally nothing is possible without them. We are also very grateful to the many members of the Wayne Morse Circle who are here with us. Circle members provide critical support for our programs through annual donations in any and every amount, small to large. Please consider supporting our work by becoming a Wayne Morse Circle member. We are celebrating our 20th anniversary this year and look forward to bringing programs like this to you for at least another 20. Without further ado, I'm going to turn this over to Judith Eisen, my colleague and professor of biology. She is Distinguished Scholar at the Wayne Morse Center this year. Professor Eisen will introduce today's panelists.
Thank you, Ellen. I'm really very excited to be part of this very interesting event. We're going to have 3 speakers today. So first, we're going to hear from Alexandra Min Stern. Professor Stern is the Carol Smith Rosenberg Collegiate Professor in the Department of American Culture at the University of Michigan, where she also has appointments in history, women's studies, and obstetrics and gynecology. Her research is focused on the history of eugenics, genetics, society, and justice in the United States and Latin America. She's the author of many books, including Eugenic Nation, Faults and Frontiers of Better Breeding in America, and most recently, Proud Boys and the White Ethnostate. She directs the Sterilization and Social Justice Lab, and she'll tell us a bit more about that today. Our second speaker will be Jada Ben Torres, Associate Professor of Anthropology at Vanderbilt University. Professor Ben Torres works in the Anglophone Caribbean, where she explores genetic ancestry and population history of African and Indigenous Caribbean peoples. She directs the Genetic Anthropology and Biocultural Studies Lab, which focuses on race and ethnogenesis, race and health, and genetics and society. Ben Torres is co-author of the recently published Genetic Ancestry, Our Stories, Our Past, which explores the scientific nature and limitations of genetic ancestry testing. Our commentator today will be Françoise Baylis, the 2021 Wayne Morse Chair of Law and Politics. Professor Baylis is a philosopher and university research professor at Dalhousie University in Nova Scotia, Canada. Her most recent book is Altered Inheritance, CRISPR and the Ethics of Human Genome Editing, which came out on Harvard University Press in 2019. Baylis is a member of the Order of Canada and the Order of Nova Scotia. She's a fellow of the Royal Society of Canada and a fellow of the Canadian Academy of Health Sciences. In 2017, she was awarded the Canadian Bioethics Society Lifetime Achievement Award. Following our speakers, we'll field questions from you, the members of our audience. We anticipate having plenty of time for those questions, so please feel free to use the Q&A button at the bottom of your screens and let us know what your questions are. We'll relay those questions to the speakers and give them an opportunity to answer them. So without further ado, I'm going to turn this over to Professor Stern.
Hello, everyone. Good afternoon, a few hours later from East Coast time in Ann Arbor, Michigan. I'm happy to be here with you this afternoon. Thank you, Ellen and the team for organizing this and Judith for those introductions. Let me share my screen as we all have learned how to do on Zoom these days, and make sure you can all see it, which I think you can. So I was very happy to be asked to participate in this panel to share my research, my historical research on eugenics and sterilization, and to connect it to thinking about contemporary issues of eugenics and scientific racism today. As Judith mentioned, at the University of Michigan, I run as a collective endeavor the Sterilization and Social Justice Lab. This lab has been up and running for about 10 years, and in it we look closely at the history of compulsory sterilization across the United States in the 20th century with a focus on a handful of states. This lab includes it's multi-institutional. We have faculty members and colleagues at other universities, postdocs, graduate students, and undergraduates. And our research is guided by an orientation towards reproductive justice and thinking about the ways in which this research can be relevant to justice issues today, very much in line with what Ellen was saying about the Morris Center. So before I tell you a little bit more about the research that we've been doing at the lab and what it's been teaching us, I wanted to do what would be the quickest overview possible, really, of eugenics. Some of you may be familiar with the term, some of you may not be familiar with the term, so doing an overview can be helpful. So this term eugenics, which some of you may have heard of in the context of, for example, my students frequently have heard about it either in the context of thinking about Nazi Germany and the Holocaust, which makes sense, or in the context of thinking about contemporary uses of genetic technologies, which Professor Benn Torres, I think, will speak to a little bit more in a little more depth. However, eugenics has a long global history and certainly a quite unfortunately robust history when we look at the United States, at the USA. So the term itself was coined in 1883 by Sir Francis Galton, who actually was a cousin of Charles Darwin. And he came up with this term with the idea that it was about improving, and these are the kind of use scare quotes, improving what would be seen as the inherent or inborn qualities of a race to improve that race to the utmost ability possible. And he used many tools to do this. He used statistical tools, he used anthropometric tools, and was very, very keen on measurement. Because if in the quest to determine who was fit and who was unfit, which was ultimately the goal of eugenics, from the perspective of eugenicists, that had to be done using classifications. So one thing to know about eugenicists is they spent an inordinate amount of time coming up with classifications to divide people and often races into different groups that were then placed into hierarchies of value. You see on the screen here, both Galton himself, there in, I guess, the middle left screen, I guess it would be if we're looking in the same direction, followed by next to him, Charles Davenport. Charles Davenport was one of the most prominent eugenicists in the United States. and with monies from the widow of a railroad magnate in 1910, created something called the Eugenics Records Office. And one of the first publications they produced was called the Trait Book. And the Trait Book was about identifying ostensibly inborn or genetic traits among different peoples and races to determine who was fit and who was unfit to reproduce, and in their view, to constitute the body politic in the United States. Next to him is Harry Laughlin, who ran the Eugenics Records Office from 1910 to 1940 until it folded because the Carnegie Foundation decided that it could no longer manage to justify the racism of the Eugenics Records Office and the kind of ideas put forth by Laughlin and Davenport in their endeavors. And finally, on the far side, you see this tree. And the tree is a very common image. It invokes kind of our evolution and Darwinian branching. And eugenicists used it because they believe that to really bring about racial improvement involved a multi-pronged approach throughout society to increase the breeding of the fit and decrease the breeding of the unfit. So the way I like to think about these different approaches of eugenicists across time, and we can think about how relevant these kind of two big categories are today, is across a spectrum. On one side of the spectrum was the focus of these white male elites, including some US presidents in the early 20th century who were concerned with the prospect of race suicide, as they would call it, to encourage white middle-class people and couples to have more babies. So on the positive side were, and some of you may have heard of them, and they sound quite comical when you think of them, but they had this sinister underbelly of eugenics, were better babies contests. So determining which was the best baby, which baby had the best heritage, which baby was likely to grow up and have the highest IQ and the best physical and mental development, as well as fitter families contests and marriage counseling. For example, it was not uncommon for eugenicists based at universities to offer stipends to young male white students who agreed to get married after graduation and had for every single child they had, or who participated in eugenic activities in some way or another. So that was the spin of positive eugenics, which was really focusing on the white middle class Americans and encouraging them to have more children in a pro-natalist fashion. On the other side of the spectrum was negative eugenics. And negative eugenics encompassed a range of different policies and measures that were enacted often on the level of the state or through laws to basically impede the so-called unfit from reproducing. One of the most well-known of these that some of you are probably aware of, and there's a history in Oregon to this, were laws that were passed to require compulsory sterilization, usually of individuals in state institutions, because they were deemed to have, and again, scare quotes, to have defective heredity, to be criminally insane, and very commonly to be feeble-minded or otherwise mentally deficient. There were other approaches as well, and they included permanent institutionalization or segregation of the unfit, as well as marriage restrictions, whereby a feeble-minded person labeled feeble-minded would not be allowed to marry a quote-unquote normal person, as well as, I would add, stringent immigration controls, such as we saw in the passage of the 1924 Immigration Act in the United States, and on the most extreme side, euthanasia, which in the United States did occur, usually in the form of quote-unquote mercy killings of babies that were deemed defective upon birth. They were, classified as having some sort of a disability or an impairment, as well as we know what happened on a horrific and massive scale in Nazi Germany. So the US, some people are sometimes surprised to learn that the US was actually at the forefront of this negative form of eugenics, specifically eugenic sterilization. In 1907, Indiana passed the world's first sterilization law, mandating that prisoners and patients in institutions could be sterilized if they were classified as unfit, feeble-minded, epileptic, and several other categories. Indiana was followed by Washington State and California in 1909. And then these laws continued to be passed all the way up until the final law was passed in 1937 in Georgia. Now, sometimes these laws were overthrown by the courts because they were deemed to be unconstitutional. But what that meant was the legislators frequently would then go back to the drawing board and figure out ways of couching sterilization laws as being for public health benefit and being for the benefit of the common good, and then put certain restrictions on them. For example, allowing patients the opportunity to appeal, even though that appeal was often just ignored, such that by 1937, 32 US states had passed eugenic sterilization laws. And this included Oregon, and I'm adding this in to have a point of reference for thinking about Oregon. Oregon is a very interesting case in the history of eugenic sterilization in the United States. The primary champion of Oregon sterilization law was Bethina Owens O'Dare. She was an active, what we would call like a maternalist. She was into maternalist politics. She was very much of a vocal suffragette and pushed for women's right to vote, based on the understanding that women had the best, white women, I should add, had the best moral understanding of society and could be the guiding hand for the improvement of Oregon and the nation as a whole. And based on this, she really pushed the legislature until the Oregon's law was passed in the 1920s. Based on that law, over 2,500 people were subjected to compulsory sterilizations in Oregon. The law was on the books until 1983, until it was finally repealed. And then the early 2000s, the governor at the time issued an apology for those sterilizations. I can talk more about that and the whole sordid history of what happened to those records, which ultimately were shredded, as much of this was discovered in the early 2000s. But the Oregon's case was one of 32 different states. And at the lab, we are looking at to have geographical representation, and also because these were the records that we were able to obtain in comprehensive fashion, we at the lab, we have compiled the records of California, North Carolina, Iowa, Michigan, and we hope to be adding Utah into the mix, covering a swath from both the South to the West to the Midwest. And we're looking at, through our research, both demographic patterns, doing statistical and quantitative analysis, and also qualitative analysis, looking at human experiences, and being really careful as we work with these records to realize that every data point that we have, every record that we have related to the sterilization of someone is actually encapsulate someone's story, and we want to humanize them as we reconstruct these histories and to treat these records with dignity as part of the past. Ultimately, over 65,000 people were sterilized in state institutions or under these state laws. And it's important to note a few things about sterilization in the US in the 20th century. First, none of the laws that were passed were ever targeted specifically racial or ethnic or religious groups. They all were written in the language of what we would call today to be disability. So a disability studies lens is very helpful in looking at the language of these laws and how they targeted those who were seen as reproductive threats due to the likelihood of reproducing, feeble-minded, and again, these are the terms of the time, which are now quite offensive and disturbing, but feeble-minded, reproducing feeble-minded offspring, or others who would be deemed unfit and defective. What happened, though, as these laws played out, is they both refracted and amplified existing prejudices, such that certain racial groups, certain age groups, and gender were targeted over time. So, for example, our analysis in California shows that Mexicans and Latinx populations, particularly younger Latinx women, were disproportionately sterilized from the period from 1920 to 1950 at rates that were, quadruple of kind of the average sterilization rates. Similarly, you will find patterns of African American women being disproportionately sterilized in North Carolina, especially as the program continues on in the context of the civil rights movement and desegregation into the 1950s and 60s. We have also found that in California, where we have some preliminary data that shows that Asian populations were disproportionately sterilized. In fact, in the 1940s, some young Japanese women who were interned at some of the camps were actually sent for sterilization and then sent back. And there is very much of a class dimension as well, both class in terms of class and immigration, so that Eastern and Southern European immigrants, those groups that were targeted by eugenically oriented immigration laws in the 1920s, were also disproportionately affected by sterilization. And there really was a class dynamic playing out among whites, where professionalized whites, like someone like Bethina Owens O'Dare, who had a medical degree, actually from the University of Michigan, I would add, wanted to police those boundaries between what they believed, what they saw as kind of white trash, often kind of rural migrants, whites who worked in factory and agriculture, and those who were part of kind of an urbanizing professional class. Now, just as Oregon's law was repealed in 1983, most of these laws were repealed, the state laws were repealed by the 1980s. There had been a transition by that time to more of a bioethics framework in terms of thinking about reproductive autonomy. However, what is quite striking is that even with these shifts that we've seen, and even with what we could call the professionalization of genetics and its bifurcation out away from eugenics, sterilization never ended and has not ended to this day. Specifically forced sterilization. So in California and throughout the country, the 1970s saw an episode of extensive sterilization of often most prominently women of color, particularly African American women in the South, Latinx women in the West, as well as Native American women, both on and off reservation settings. In addition, as recently as the period between 2006 and 2010, about 150 women were sterilized in non-consensual procedures in 2 California women's prisons. This, the information about this turned up in a journalistic expose in 2013, resulted in a state audit, the passage of a law in California, prohibiting sterilizations in California state prisons. And there's an ongoing attempt to pass a bill that would provide reparations both to the women sterilized in those procedures and for any remaining survivors of the earlier period that I talked about in the research that we do, that dates from the 1909 to the 1970s. And I will just wrap up now by, saying it is very much true that this type of reproductive abuse continues to happen. And most recently, some of you may be aware of the whistleblower who worked at the Irwin County Detention Center in Georgia, who alerted lawmakers and the media to the fact that women, often Central American detainees, were being forced into procedures, often in the context of surgeries for things like gynecological cysts or other types of conditions. They were brought in and they were subjected to unwanted sterilizations. The ramifications and the aftermath of this situation continues to play out. There are advocacy groups that are both protecting the whistleblower and pursuing justice for those who were harmed by these policies. I'd be happy to talk in the Q&A about what were the conditions at Irwin that gave rise to this horrible situation, and how does understanding what happened in 2020 relate to understanding what happened in all these different US states in the first half of the 20th century. But I've run out of time because there's so much to say about this topic, and I will stop sharing my screen and hand it over now to my co-presenter, Professor Ben Torres. And you're
Okay, I want to say, Alex, thank you for that great talk. It was very informative and provocative in terms of thinking about how these things continue to happen today. So before I begin, of course, I want to thank Judith and Ellen for pulling this all together and for allowing us to have what I hope will be a really interesting conversation. So I'm going to begin. Let me share my screen, and I'm going to focus on genetic testing and specifically direct to consumer genetic testing. So direct to consumer genetic testing, these are tests that are directly available to the consumer without the intervention or even the interaction with a medical professional. These tests are provided by a number of companies, some which are a little bit more reputable than others. But these tests allow individuals to purchase a kit at a grocery store or a pharmacy, and then for a fee, send their DNA back to be analyzed. Some of these tests can tell us about things like our ancestry, potentially our genetic risks for certain diseases, and give us a sort of a general idea of how we may be related to others others that have been tested. And this is of particular interest for people that are like to use genetic data to enhance their genealogical research. So these direct-to-consumer tests actually have been available since the late 1990s, with the first and actually still most purchased tests being paternity tests. With these tests, as I mentioned, you submit your DNA to the company for testing, usually without the intervention of a medical professional, including a genetic counselor. From there, the company can then send back the results to you, and you can decide how to engage or interact with these tests. So in my own sort of work and day-to-day life, I've noticed that these tests are actually increasingly available at local retailers. Here's an image actually I took at a Target store. I think I was in Boulder, Colorado. This was 2 summers ago. So you can find these DTC tests at your corner store pharmacies, general stores. I've even been to bargain basement stores and seen some of them on sale. The appeal to these sorts of tests is that the consumer can learn presumably novel information about themselves and their families using something that's as personal and as intimate as their own DNA. In this case, direct-to-consumer testing, it can provide a sense of supposedly objective way of learning about yourself and your relationship to other people. Now, I say supposedly objective because I like to make the point that genetic testing, like any science, is still a product of culture, right? So the questions that we ask, which can shape study design, but the questions that we ask and the answers that we get are always filtered and bound through the ways in which we understand and engage in the world. So in my own work in the Caribbean, and more recently with people who have taken genetic genealogy tests, or these genetic ancestry tests, I've learned that sometimes people will take these tests with the hope of learning more about their families and their family histories, but at times, these tests actually can lead to more questions than answers. So before I discuss the genetic potential of these direct-to-consumer tests, I actually want to spend a moment really discussing how these tests work. So I've separated it into 2 very, very broad categories in terms of what's available to purchase. So the first are these genetic ancestry tests. And the basic premise of these tests is that the consumer will purchase the test, send their DNA in, and their DNA will be compared to different reference groups. And these reference groups consist of different populations who are believed to have generally stayed in the same geographic area and have not, in theory at least, mixed with a lot of other groups. Again, I say that's in theory because we are, as a species, really good at making babies with one another. So we move around and we have offspring. So again, with these ancestry tests, your DNA as the consumer would be compared to these reference groups. And the proportion of DNA that you might share with these reference groups can be calculated to estimate your ancestry, or a probabilistic assessment of your relatedness to these reference groups. Okay? There are a number of caveats with these ancestry tests. There are some ancestry tests that will focus only on the maternal lineage. Okay, so to learn specifically about the maternal ancestry, we look at a specific part of the genome known as the mitochondrial DNA. So the mitochondrial DNA is actually a special type of DNA. It's not located in the nucleus where the rest of our genetic material is, but instead it's located in a little organelle that's in our cells, and this organelle is responsible for energy production. This organelle also has its own DNA, okay? And this DNA is actually inherited from the mother, and only females pass it on. Okay, so if we look at our pedigree here, I could focus on the bottom first, where you've got a son and a daughter, and they have their parents, the mother and the father. So with mitochondrial DNA, I mentioned that it's inherited from the mother. So this is illustrated in this figure as this little red circle. Okay? Mitochondrial DNA actually does have a circular formation. And you'll notice that it's the same between the siblings here, and their mitochondrial DNA is the same as their mother, which is the same as her mother, which is the same as her mother, right? So when we look at mitochondrial DNA, we could say something about the maternal lineage, okay? It's pretty low resolution analysis is what we call that. Alternatively, if we were interested in looking at the paternal lineage or paternal ancestry, we can look at the Y chromosome. And the Y chromosome contains all the genes that make chromosomal men men. Okay? In this figure, it is illustrated by this little blue chromosome. Okay? And you can see, number one, that only the son has it. Again, it's because he's chromosomally male. And his Y chromosome is the same as his father, which is the same as his grandfather, which is the same as his great-grandfather back in time. So we can use the Y chromosome to learn about the paternal ancestry. And both, as I mentioned, mitochondrial and Y chromosome DNA, we consider these to be low resolution analyses because they don't tell us a lot about a person's overall family history. And they can also give us an idea of sort of deeper ancestry, more so than more recent ancestry. Okay. There are several other caveats with both mitochondrial DNA and Y chromosome DNA that make it really interesting to look at, not only in individuals, but in populations, to get broader ideas about how human groups have moved across the globe and how they've interacted with one another. Regardless, these parts of the genome are used in genetic ancestry tests, and they do provide some insights into a person's sort of deep ancestry. If you are taking an ancestry test and you are interested in having a more general overview of your family tree, then you want to use the nuclear part of or the DNA that resides in the nucleus of the cell. So you want to look at DNA that's biparentally inherited. Okay, so inherited from both the mother and the father. And in our illustration here, this is what we would say are the other chromosomes, okay, or autosomal chromosomes. And here you can see that these are represented by these multicolored chromosomes, okay? And what this indicates is that this is DNA that these individuals have inherited from their parents, okay, from both of their parents. So by looking at these autosomal markers, these autosomal DNA, we get this overall picture of the family. One other sort of important point that I want to explain is why these chromosomes down here are mosaics, yet in the great-grandparent generation, you see these solid lines. Okay? This is for illustrative purposes. In reality, everyone's DNA actually has this more of a mosaic look. What this image is trying to show is that through the process of recombination, so this is something that occurs with gamete formation, our genes are actually shuffled between our chromosomes. So if we look, for example, let's see, we'll start with the great-great-grandmother. You can see that her parents have kind of this teal and red series of chromosomes. What this grandmother here has inherited is one from her father and one from her mother, okay? What she's going to pass on to her offspring would be genes that have been shuffled between these chromosomes. And you start to see that represented here. Okay, so you start to see stripes. And this is just showing you through the process of recombination, of gene shuffling, that this individual, this mother here, will share some DNA with her grandmother, with the grandmother, and in this case is her mother, and the grandfather or her father. Okay, so this happens every generation. Your genes get shuffled and then passed on. So when we look at the autosomal markers, what we're actually seeing is contributions from more of the family tree. Okay, so depending on the sorts of questions you might have as a consumer, you might want to look specifically at your maternal ancestry. If you're a chromosomal male, you'll want to look at your paternal ancestry. And then for your overall family, you'll want to focus on the autosomal markers, right? Because that will give you a more overall general view of your family. Now, just to make sure that I am clear, all of these ancestry tests actually have some important caveats and limitations to them. So if we were focusing only on maternal ancestry, okay, as I mentioned, it's low resolution. Everybody with an X over it, we don't really know anything about these individuals. And actually my slides have shifted over, but you should see X's over here as well. The only people that you'll be able to see with the mitochondrial DNA would be the maternal lineage, okay? For the X chromosome, okay, so if you're interested in looking at the paternal lineage, again, you don't know anything about individuals on this side of the family tree, okay? So you wouldn't know anything about, let's say, the mother's father, etc. All right, because they don't have Y chromosomes. So those are a little bit more low resolution. For our autosomal tests, the real limitations start to come up with comparisons to reference populations, okay? And this is particularly problematic for comparisons to populations that maybe have a more complex recent history, okay? Because of my work and my interest is applying these technologies to people in the Caribbean and folks of African descent. One great example that really illustrates this problem of reference populations is thinking about trying to use DNA to figure out the origins of African ancestry, like who was taken from where in Africa as a result of the slave trade. Part of the issue is identifying appropriate reference populations. In particular, there are groups of people and ethnic groups that didn't really exist or don't exist in the same form now as they may have 200, 500 years ago. So it can be very difficult to draw a line between the present and the past, okay? So this general problem with these autosomal ancestry tests, a lot of it really does lie in the quality and the quantity of the reference groups. These commercial companies are increasingly better at advertising the numbers of reference groups that they have. But this can actually make a big difference in really assessing the quality of ancestry results. In a conversation I recently had with individuals who were taken ancestry tests, a lot of them were really perturbed by the fact that their ancestry estimates were changing. And what was happening was that these companies were updating their reference populations and then recalculating ancestry estimates based on the newly revised reference population. So in effect, they were giving the consumers sort of better answers, but people were really thrown off by seeing these numbers change. Okay, so these are genetic ancestry tests. With some commercially available services, you can also get an idea of your risk for different diseases. Okay? And where this information comes from is basically a series of genome-wide association studies that have been done. Okay? So researchers essentially will be looking for different genes and genetic markers that are implicated in a disease. So I want to talk very briefly what these genome-wide association studies are, and then use that to help everyone sort of think about what it is you're actually purchasing when you buy these tests. So the aim of GWAS, or a genome-wide association study, is to detect associations between allele or genotype frequency and a trait status. Usually that trait status is a disease. So essentially these sorts of tests let scientists find places in our DNA that can be associated with particular diseases or traits. So the way this works, if we look at this image, these studies will typically start with a case control study design. Okay, so you'll have cases, so people who are affected or who have the disease of interest or the trait of interest, and those who do not. So we're actually just going to focus on this column for now. Okay. What they'll do is they will look, assess using a SNP array. So these are like little chips with millions of regions of the genome that are targeted. Okay, so they'll look and see what the sequence is at many places in the genome. Okay, and they're doing this using something known as single nucleotide polymorphisms. So that's this, SNPs. Okay, so they're little regions in the genome of interest, and they'll genotype this in the cases and the controls. And then they'll do association analyses, okay, to statistically detect if there are differences in the types of SNPs that show up in cases versus those that show up in controls, okay? And what this is showing down here, this dotted line, this is a Skyline Plot, is basically our line of significance. So when you see any sorts of dots up here, this means that these SNPs, okay, are of interest. They are potentially associated with the trait of interest. Okay, you've got some in, for example, here, this is from chromosome 11 that's approaching significance but is not significant. It looks like for this example, there would be several SNPs on chromosome 1 that would be of interest. In this column, this really just describes another way of doing a genome-wide association study. Here you've got an unselected sample, so they're not necessarily split into cases and control. And what they're doing here is a whole genome sequencing. So they'll sequence everyone's genome, run similar analyses to figure out where associations are with the different SNPs. Okay? One important caveat with these genome-wide association studies is that they don't assign causality. Okay? They are association studies. So they'll just tell us that these particular markers are associated with a disease. There are additional experiments and tests that have to be done to assign causality. So when you take these genetic ancestry tests and you're trying to learn more about your risk for diseases, what these companies have done is basically reviewed the literature, and they're presenting information in terms of the significant findings from different GWAS studies. And there are several caveats to really interpreting and thinking about those. I have some of these caveats listed here, and just for the interest of time, we can't really go over it. I do have the citation at the bottom if anyone is actually interested in learning a bit more about genome-wide association studies. So there's a couple of limitations that I think are super important to consider, particularly because this information is used and marketed as being informative about people's risk for particular diseases, right? So one of the limitations of GWAS studies is that they don't actually explain a large fraction of why a disease may or may not be heritable. Okay, And so when I say heritable, I mean it's actually a factor we can calculate that shows how much genetics or genetic inheritance actually influences displaying the particular phenotype or the disease. So these GWAS studies really just tell us only a little bit about the heritability, right? So there's some missing heritability in this. In addition, because of the we call it genetic architecture of diseases, there's actually very limited clinical predictive value for now. And this may not be the case forever. And when I talk about genetic architecture, I mean the genetic underpinnings of a disease. And there's different ways that diseases can manifest genetically. Sometimes you can have these variants, which are not very common in populations. We call them rare variants. And these rare variants, again, not very common, can have little effects. And with enough of them, it can manifest in disease. Sometimes diseases have a different genetic architecture where you have variants that are actually pretty common in populations, and they also have little effects. Okay? So depending on the genetic architecture of the disease, the way the genes work in tandem to actually cause disease, it can be really difficult to use these statistical methods to detect them. What that means in the end for the consumer is that even though you might have a particular variant that has been associated with a disease, there's still a possibility that you will not have that disease. This is not the case for every sort of genetic disorder, but for complex diseases, ones that affect lots of people, things like cardiovascular disease, diabetes, for example, we are learning increasingly that these sorts of diseases are pretty complex at the molecular level. Okay? So while there are my point in sharing all of this for you is not to just sort of trash GWAS studies. I don't want to do that because, as you can see on the screen, there's a lot of reasons, a lot of things we actually have learned as a result of GWAS studies. But my point in really sharing this with you is that these studies have to be very carefully interpreted, okay, and utilized in ways that make sense for an individual's disease risk. And as I mentioned, often these DTC products are available without any sort of interaction with a medical professional, with a genetic counselor who understands more of the statistics and disease architecture, genetic architecture to explain this. So the results from these GWAS studies are not a one size fits all. Okay? There's actually a number of statistical, biological considerations that have to be accounted for when we think about how applicable the results are to consumer bases. So increasingly, companies that market these sorts of tests, they do offer some information about how to interpret the test. Some also are beginning to offer information about finding a genetic counselor or someone who could help explain your actual risk. My understanding is that med schools are beginning to have to engage with this so that physicians who potentially can encounter patients who have done these tests and want more explanation, they've got to be able to talk to them about this. But arguably the provided information from these companies is still pretty specialized, right? And for the average person who's engaging with this type of information, there's still a lot of room for misinterpretation. And this can have implications about how people use and engage with this technology. So beyond the interpretive limitations and the caveats of genetic testing, as I encounter the ways that DTC is marketed and used and consumed, I think overall I am concerned by an underlying premise of genetic determinism. Okay? When I say genetic determinism, I'm referring to the belief or the understanding that one's fate is, or at least largely, is at the mercy of one's genetic code. Okay? For example, I see elements of genetic determinism in critiques about genetically modified foods. Okay. Some of these critiques posit that by changing or altering the genetic sequence of an organism, you are actually altering something essential about that organism. Okay, so I also kind of see these similar tropes in terms of genetic determinism in some of the ways in which personalized genetic medicine is discussed in public forums. Okay. In this case, people might be out to beat their genes, okay? So they might choose not to eat this or might want to eat that, or engage in particular activities. And the idea is that they're trying to beat fate, okay, beat their genome. So as scholars, as different stakeholders, I think that as we encounter these technologies, it's really important to consider both the advantages and disadvantages of really engaging with consumer-driven testing. When the sorts of genetic tests that are available are really driven and focused on what companies are willing to invest in and develop, and that's based on what they think will actually sell, we have to think, will this lead to issues of injustice, right? Where there's not equitable access to this sort of information, or equitable technology, let's say, or questions that are being asked to serve all members of our society. Also, I think we have to also consider the very strong potential that the ways that genetic tests are interpreted can lead to reinforcing wrong ideas. Things like, again, genetic determinism, or even race as biology. And this was mentioned a bit by Alexandra in the previous talk with the origins of American eugenics. How are these things to be combated when we have companies that market their tests in very specific ways? Finally, I also think it's really important to consider what people may want to do with the information from genetic testing. And again, my role is not to impose my will on anyone, but I'm hoping to provoke conversations about what these technologies give us the power to do and how it might shape our own lives now and in the future. I am interested in provoking conversations around how we as a society might come to terms with the possibilities and the power to actually shape the genetic makeup of future generations. How do we weigh individual choice versus a shared sense of morality surrounding genetics? So some people actually are already grappling with these types of decisions and these types of questions. Currently, there are fertility companies that offer pre-implantation genetic testing for couples that are undergoing IVF. So this is a screenshot that I took of one such company. This is the Fertility Institute. Okay. So here, this particular company is offering a variety of services to customers that purportedly help people have the healthiest babies they can, in addition to having parental preferred choice for certain traits. So in this example, the company is offering to help parents pick the eye color, and according to the website, soon hair color of their offspring. Okay. In their website, and you do have to do a fair amount of digging to find it, but they're very clear that they are not modifying the genomes of embryos, but instead they're testing the mother and the potential father to see if they already contain the variants that result in blue eyes or green eyes or what have you. So they'll screen the would-be parents, and if those variants are present in the would-be parents, they'll then screen any embryos. And this is considered an add-on feature for those who are already doing IVF. According to this website, the success rate in the past hasn't been that high, but they've more recently been in the process of revising their protocols. And now they are advertising a 90 to 95% predictive for eye color using their particular technology. So assisted reproductive technology companies also often offer genetic screening to aid in things like family balancing, okay? This is another way of saying sex selection, okay? So they can choose to, or couples can choose to implant embryos that have the chromosomes, the sex chromosomes that they desire. So this is a much broader topic than I actually have time to go into. But again, I'm just sharing this with you to show you that this is not the future, that this is now. And these are parts of conversations that we should be having. As we become more immersed in the present where eugenic choices are at hand, I want to encourage and again provoke conversations so that we can think about how such tools might change our society. In particular, our ideas about differently abled bodies. So in the last year, at the end of last year, I think this is from December, an interesting article was published in The Atlantic by Sarah Zhang. So in this article, she describes the outcome of widespread prenatal genetic testing. So Zhang writes, and I'm going to quote this here, in 2004, Denmark became in 2004, Denmark became one of the first countries in the world to offer prenatal Down syndrome screening to every pregnant woman, regardless of age or risk factors. Nearly all mothers chose to take these tests. Of those who get a Down syndrome diagnosis, more than 95% choose to abort. She goes on to note, yet a gulf seems to separate the publicly expressed attitudes, so these would be attitudes about people with Down syndrome, and private decisions. Since universal screening was introduced, the number of children born with Down syndrome has fallen sharply. In 2019, only 18 were born in the entire country. About 6,000 children with Down syndrome are born in the US every year. That's the end of the quote. What Zhang's article goes on to discuss is that since the community of people with Down syndrome is diminishing, so are the resources, including social support networks for families and caretakers. So from this example, you can see that the accessibility of genetic testing, it has significant impacts that go well beyond the individual and individual choice, but instead can also shape the demographics of future populations. So I kind of want to wrap up my time with a book that I actually used in a course I taught recently, like last semester. So in this course, I explore with the students the potential for genetics to alter some of the most fundamental aspects of being human. Right? So this can include how we might use genetics to say something about our identities, whether it's an individual identity, our kinship ties with a particular community. We also consider how DNA or genetics is used to alter our food. So how we acquire food, how we produce food. We also consider how assisted reproductive technologies, okay, these considerations of the genome, how that potentially also affects the ways in which we have babies. So I used this book by Jamie Metzl called Hacking Darwin, and my students overall found it to be quite provocative, and we actually had a number of lively discussions. The central argument in the book is that the power to alter our genomes is inevitable. However, how we as a society choose to wield this power is not yet decided. Metzl hopes to initiate the types of conversations that will shape how these technologies are employed. Now, from the perspective of a professor as an instructor, I thought the book was actually pretty good at discussing the scientific possibilities of eugenic futures. But then I thought it failed, really, at seriously considering the human, social, and cultural costs of these futures. The book was missing inputs from the humanities, as well as from voices from the communities that will be affected by these technologies. And it's my hope that forums like our current session will be exactly what's needed as we all are directly or indirectly have these opportunities to manage the impacts of increased genetic engagement. So I'll stop there, and I'm looking forward to a great conversation. Thank you.
Well, hello, everybody. You have just been treated to a whirlwind tour of things genetic. And my challenge is to really try to take the comments that you've heard from both Professor Stern and Professor Benn Torres and try to pull them together for you in a way that you can see how the one feeds into the other and take us right up to today with the range of reproductive technologies that are available to us to make choices about. So let me go back a little bit and start at the beginning with some of what you heard with respect to compulsory sterilization. And for me, when I hear those stories, no matter where they're grounded, what really comes to the fore is the need to acknowledge and deal with the fact that racism is deeply embedded in healthcare. And I say that because when you look at some of the choices that were made with respect to sterilization, while they may have been justified or explained in the context of a disability lens, you heard the language of people who were feeble-minded, people who had mental disabilities, again, the language of the day, But when you actually start looking closely at who some of these people are, you're actually able to see, quite frankly, stark racism. And I want to make that claim in the context of my country, Canada, so that I'm not pointing fingers at anyone else. And just to say that from the 1930s or so to the early 1970s, we had 2 provinces that actually had legislation on the books permitting compulsory sterilization. And the people who were the victims of that legislation were predominantly Indigenous women. In some years, as many as 95% of the sterilizations were performed on Indigenous women. And finally, in December of 2018, a class action lawsuit has been started to try to address this huge injustice. And so one of the things I want you to think about is the ways in which genetics are presented as somehow neutral, factual data out there in the world, and that we need to understand that we bring to whatever that data is a number of evaluative perspectives, and in this case, evaluation about certain kinds of traits. But it's not just evaluation about those traits, it's actually assumptions about what those traits are and how they are or are not connected to genetics. So one of the things that I think is really important to appreciate is that in those early days, and I think for some people still today, eugenic practices ultimately are the enactment of false beliefs about things like what's the cause of poverty, of crime, of illness. And somehow the idea was that this was in the genes. And so the only way to deal with this was to make sure that those genes were not being reproduced. But what happens in that context is that you're actually placing responsibility for social problems on individuals without any justification for thinking that that's where those challenges rest. A real failure to appreciate social responsibility for the outcomes of these behaviors. And so for me, one of the things that behooves us to think about at all times is gene-environment interaction. So many of us pay attention to genetics, and there's a wealth of information there as we continue to unpack our genomes. But we need to understand that genes are not working in isolation. They're working in environments, and those environments are really complex. It's everything from what we eat to what we breathe to, I would say, who we interact with. That's all part of our environment, and that affects the expression of whatever genes we do or don't have. So one of the things that I think is really interesting then to appreciate is how that sometimes gets lost. And you heard about that with reference to genetic determinism and somehow thinking that there's a gene for everything, a gene for this, a gene for that. And many of you might remember hearing about, the gay gene, the gene for obesity, the gene for violence, criminality, aggression. And not a word of a lie, you can look this up on Google. 2 days ago, my cousin who lives in England sent me an article saying, look at this, there is such a thing as the gene for bargain hunting, and we both have it. And you can laugh, and I did when I read that, but I thought to myself, what's gone wrong where we think that we're going to explain behaviors that clearly are socially informed by looking for some kind of genetic causation? So I think that's one of the things to take away. What is it that we imbue with when we think about genes? What is it that we sort of assume can be found in them and then can make certain claims about objectivity, when in fact it's all kinds of personal and social biases that are brought to bear and that inform that assessment. The other thing I want to talk about is this idea about somehow with direct to consumer sales of genetic testing kits, that in fact, then in some way, shape, or form, people are able to control how they're going to use information that's available to them, and in some sense, if you will, not be subject to other people's interpretations of their genetic material, except insofar as it might have information about their health. And one of the things that was very interesting to me when I was looking at this in general was to try to understand, why do people do this? Why do they go and pay anything from $100 to $250 to get this additional information? What are they looking for? Well, some people say that they're looking to find out where they come from. And in some cases, that means they're hoping to confirm some kind of an origin story, or they're hoping to reconstruct a family history, or they're hoping to find the geographic origins of their ancestors, the search for a homeland. Well, I'm going to say to you, as you've heard from all the science that was presented earlier, you may not find that. Another thing that people say they're looking for is they want to find out who they are. They want to find out their ancestors, their race, their ethnicity. And in some cases, people are really specific. They say things like, I want to find out my ethnic identity. Am I Haida or am I Maori? I want to find out my religious identity. Am I Parsi or Jewish? I want to find out my caste identity. Am I Brahmin or Dalit? And I think, again, I would say back to you, you're not going to find that answer. I'm not saying those are not worthwhile questions. I'm just saying, why would you think you'll get that from genetic testing? Another thing that people say is, I want to find my family. And in that case, they're looking to identify biological relatives. And we hear that, for example, from people who have been adopted. We also hear that from people who are born of assisted reproductive technology. And in this case, they may actually be amongst those who can get the information they're looking for. But it may turn out not to have been information they wanted at the end of the day. And so one of the other things then to think about is a more pragmatic context in which people are looking to genetic testing to actually access citizenship rights. They need to be able to prove a particular kind of heritage in order to get access perhaps to funding that's been set aside for a particular ethnic group in terms of reciprocity. And lastly, a lot of people say they're doing this just because it's fun. It's a hobby. And again, maybe you'll have fun, but you might learn some things you didn't think about or want to know, like non-paternity. Or maybe you're going to find out you have something like 50 half-siblings if you're a child of reproductive technology. And so one of the things to be acutely aware of is, what is it you think you can learn from genetics? And what is it that you might learn from genetics? And which of those things did you think you were going after? And that brings us to a really important ethical issue, which has to do with informed consent. What is it that people understand, especially if this is not mediated through a healthcare provider in some way, shape, or form? I want to say, though, that part of the reason I say you're not going to find some of the answers that some people think they're looking for is because, again, what you're doing is you're actually reifying the gene, and you're thinking that the answer to these questions, many of which ultimately are about identity, can be found in biology. I argue that that's not where your identity rests. Your identity rests in yourself. It's a function of a narrative. It has to do with history, memory, belonging, recognition by others. All of these things are very complicated and social. They are not reducible to our genes. And so in that context, I want to bring this to a close so that we can open it up for conversation by saying that it's some of these beliefs that we sometimes bring to the choices we make with respect to the increasing array of reproductive technologies. And when we actually get to the point, which I think we're on the cusp of doing, where we're not merely selecting, as was pointed out earlier with respect to things like eye color or hair color, but we're actually trying to design and to make a modified species, what are the racial and other biases that we might bring to that project? And that's really where we come full circle. If you understand how in the past we have used what is now considered mundane technology to ultimately harm segments of society through something as basic as compulsory sterilization, controlling their reproductive capacity, what might we do in a modern context with much more sophisticated technology and not having the state impose those choices, but having re-educated people into thinking they're making independent, informed choices that ultimately are harmful because of the false beliefs on which some of those choices will be based. And with that, I'm going to invite everybody to come back, join us, and hope we can start an interesting conversation.
I want to thank all of our panelists. I think this has been very, very provocative, very, very interesting. There's a lot of interest from the people who've been listening. We have quite a number of questions. So I'm going to start right off the bat for one question for Dr. Stern. And can you say if there's been any modern eugenic rhetoric from Western supremacists like the Proud Boys?
Certainly. As Professor Herman mentioned, and also you mentioned, Professor Eisen, in your introduction, my most recent book is entitled Proud Boys in the White Ethnostate, and looks at the most recent manifestation of white nationalism in the US over the past decade. And in looking at that, one of the key components for me was tracing the continuous thread of eugenic thinking from the early 20th century to today, with its white supremacist components, when motivated in large part by demographic anxieties. So if you actually begin to look at, for example, what is the motivation behind creating a white ethnostate for white nationalists? It's really a eugenic project. It's about creating a territorialized space that in their desire would be 90% white, and in which breeding would be largely choreographed and orchestrated by the putatively male leaders, and it would kind of solve all of their problems. At the same time, what motivates many white nationalists and those on the far right today is the sense that the demographic clock is ticking, not in the direction that they would like. That we know the United States, come 2040, 2050, depending on census projections, will be a nation in which there is no ethnic or racial plurality. It will become, as some say, it will no longer be a white majority nation. And this incites great panic among white nationalists today, and is one of the reasons for their deep concern about immigrants, for their continued anti-Black racism, and so on. So I could point out, connect the dots in a variety of different ways, but I would say that certainly white nationalists have ridden for a long time the bandwagon of eugenics, and they don't delve so much into the complexity of all of the technologies that Professor Benn Torres mentioned, but occasionally they'll bring them in when they understand them sufficiently to do so, at least to how IVF and so on could be used. And there are several European far right thinkers who like to talk about the idea of a retro culture that would bring back what they view as kind of the white nostalgia of the 1950s and mesh it in with the contemporary genetic engineering of today to reproduce what for them would be this wonderful utopia, and for many of us would be definitely a dystopia.
Thank you, Dr. Stern. I'm going to move now to a question for Dr. Benn Torres. Can you speak more about the relationship between direct-to-consumer ancestry testing and the false conception that race is biological?
Yeah, that's actually a great question. One of the issues with the marketing of direct-to-consumer genetic ancestry tests is specifically the ways in which sometimes these things are marketed. They will ask in bright letters, what is your race, what is your ethnicity? And they presumably can tell you if you spit in a tube and give them some money for it. The issue is that race is not biological. And I say this is kind of contemporary anthropological thought on the subject. Our ideas of race are what we call, it's a social construct. These are a series of ideas that we have made up and impose on our society to kind of organize and think about people categorically. The traits that you might go to think about what race a person belongs to, these things actually change over time, and they change over geography. So for example, if you were to travel to Brazil or to Puerto Rico, there's a different sort of cultural understandings of what consists of race. This is an indication to you that race is something that is not biological, but is a series of collective understandings about human variation. When I say this, it's sort of off-putting because I'm not at all saying that human variation is not real. Human variation is real. You can look at just this panel and you can see the many ways in which we're different. What I am saying is that there are cultural rules that sort of surround the value and the meaning that we give to those differences that we see. And those values and meanings actually change over time and over space. And that's why we think about race as being a social construct, as being experiential. And no genetic ancestry test can tell you that. Oftentimes in my classes, I'll show my students my own sort of genetic ancestry results, and you will see that I have European ancestry. And then I ask them, like, if I go missing, please don't tell the police you're looking for like a white woman. You're not going to find me. Tell them, you know, how you understand me to be. And in that case, I'm asking them to tell like the police about my social identity. Okay? These again are cues that we use socially to identify ourselves as different groups and belonging to different communities. However, this is not found in our DNA. We can say something about our ancestry. All of us probably have people in our family tree who might self-identify racially differently than we do. Okay, so again, it's not something that a DNA test can tell us, but instead race is more of a social construct and a collective understanding of how we understand human variation.
Thank you for that answer. So I'm now going to put out a question that maybe Dr. Baylis would like to start with, but maybe everybody would like to address. So what practical steps do you propose that can help involve and educate the public about the ethical as well as scientific issues connected to consumer genetic testings and its potential uses?
Well, I think there's a number of things that I think are extremely important. I made very quick mention of informed choice. I think that people are capable of making a choice to do ancestry tracing, and they may genuinely think that it is just for fun. And we do know that some people who get their results find it entertaining. But what I'm worried about is these are consent forms that are extremely long and complicated. In some cases, they're online. And over time, we have been trained to just hit accept, accept, accept so that we can get to the page we want to get to. So many people don't read those consent forms. And one of the reasons for reading the consent forms is to actually understand what's going to happen with your information. So it's not only what the information is going to mean for you, but how that company will use that information. You've already heard from Dr. Benn Torres, the reason that, you know, people's profile, if you will, shifts is because they have to recalibrate as they get more and more data into their databases. That more and more data is more and more people paying them $100 to give them their information to, in fact, make them richer and to make their data more and more valuable. And those companies can then turn around and sell that data. Did people understand that when they consented to what might have been for them just a lark? I think that's one of the first things. Informed choice is critically important for people to understand what it is they're participating in. But the other thing that I try to explain to people, and I very often in a teaching context use myself as an illustration to try to disturb people's assumptions. And one of the ways in which I do that is I say to people that I claim a Black identity. And people look at me and they're shaking their head, right? Why? If I took a DNA test and I had some proof, would that make it true? When I turn around and I tell people part of the reason I know with confidence that I'm Black is my mother is Black, and she is genetically my mother. She's from the Caribbean, and they might say, okay, well, still a bit odd. And then I can say things, oh yes, she used to be called a freckly banana. That was the color coding that was used within the sort of spectrum that you would find in Barbados. But what really shocks people, and the only reason they give credence to my claim, is because my mother is the first successful case of racial discrimination in employment in Canada. So she actually had to prove, and she was asked in court, are you a Negro? So what would it mean for me to say that I'm not Black and to disavow my mother? And yet what's really interesting is that when I say that, I can't tell you the amount of responses I get that are just evidence of racism. And the most stark one is, oh, well, you're not like all the other ones. You want to say, what does that mean? What assumptions are you bringing to bear? And in that context, I think it's those kinds of conversations that you want to have. You want to ask people, what's family? Family is not blood. I have all kinds of people that I call aunt and uncle who have no genetic relationship to me, but they're family in the meaningful sense of the word. And that brings me back to the reproductive technologies. If we think that the only families that matter are those with genetic ties, we're actually undoing a lot of very important work that we've done to recognize the importance of social relationships. So I think, to sum it up and to give others a chance to respond, I think a part of me wants to make sure that people are educated enough that they understand what it is they're participating in, so that it was actually a meaningful choice. They knew why they were looking for this information. They knew what the chances were of getting this information, and they knew all of the other uses that could be made of their information. And then the other ways in which I want people to be sort of more thoughtful about what they're doing is I want them to actually explore, what is it that you think you're going to find? What is it that you're looking for? And is this the best path to get where you need to go? Do any of my colleagues want to weigh in?
Sure, I'll weigh in. I think it's also important to consider if you are going to, purchase a test and maybe see what you can learn, you need to realize that it's not just you, that your family is going to be implicated in this as well. And for some people that doesn't matter. The family actually might be interested in all aboard. But for others, there might be a little hesitancy for having that information out there. I will also add that with a lot of these direct-to-consumer testing companies, you are able to download your data. They'll give it back to you, your raw data, and then you can upload it to other sites. And when you upload it to third-party applications, I think at that point, you're really opening yourself up to uninformed uses of your DNA. Within the context of DTC companies, there are things you can opt in and opt out of. So you have a little bit more control as to what happens to your data. But if you pull that data down and upload it somewhere else, and people do this, there's something known as GEDmatch that allow people from many companies to upload their data. And it can really expand, if you're doing genetic genealogy, it can expand your search for finding relatives. So in that regard, people love it. However, I think it was, as was the case, it was last summer or maybe the summer before, various law enforcement agencies were able to search those databases. And they were actually able to locate a serial killer. And I think all of us would agree that this is probably a good thing. We don't want serial killers roaming the streets. However, nobody consented to having their DNA used in that way. Okay? This is not an original usage for this database. So you have to think very carefully as to if you're going to use these third-party applications, are you open to the possibility of your DNA being used in this way? And again, it would be part of a broader conversation that we would need to have in terms of thinking about, well, if we have these technologies available, how should we use them in the context of law enforcement? How can we do this equitably if we're going to do it at all? And I think I'll kind of stop there so we can get more questions.
I can just add to this that, what I discussed in my talk focused primarily at looking at the long era of state mandated reproductive control, where it was state laws and state institutions, whether it was mental hospitals in California or prisons in California or detention centers in Georgia. These were federally and state controlled facilities where individuals were deprived of their reproductive control based on prejudices against people with disabilities and racism, etc. It's really interesting. I always go back when thinking about the history of eugenics and genetics to the period of the 1970s and 80s, when there was something of a transformation away from and a rejection of this kind of state control to something that resembles what we would look at as contemporary bioethics today. And I'd love to hear Professor Baylis's thoughts on how far this goes, but a recognition of reproductive autonomy. So for example, because of all of the cases of reproductive injustice involving sterilization abuse in the from across the 20th century and really culminating in the 1970s with these high profile class action lawsuits and hearings that were held in the US Congress, the laws actually changed, whereby for someone to be sterilized, especially if they were using Medicaid or federal funding, a waiting period was mandated. Forms had to be translated, consent had to be given. Now, that was never absolute. Those safeguards, we could argue, did not go far enough. There were many cracks in that architecture, let's say, but that did also lead to a period where we moved from, eugenics, and let's call them these proto-eugenic technologies, moved from kind of the state, broadly speaking, to a more commercialized and a kind of beyond the state. So one of the kind of where we see the prospect of eugenics today certainly can be in the context of kind of, state control, but it is just as much embroiled with these commercialized technologies and these privatized technologies. One of the books that I've written is on the history of genetic counseling, and genetic counseling is a field that has been tangled up in eugenics in complex ways and been trying to free itself from that kind of that eugenic shadow. It is also a field that it doesn't have the stature of, some of the PhD and MD geneticists, and has been more beholden to some of these commercial genetic companies. And there's been a lot of debate among genetic counselors of the extent to which participating in kind of advertising by genetic testing companies is capitulating to their kind of implicit eugenic rationales or assumptions and so on and so forth. So I think that broad sweep of, kind of from the state to kind of to the more commercialized landscape, where a lot of the ethical questions are being raised in the context of this hyper commercialization. And it's really the selling of products through, you showed some of them, Professor Ben Torres, these really like flashy websites and brochures that can pull people in because they feel like it's going to give them information, solve health problems, help them figure out their identity, and so on and so forth. So that's kind of, in a broad sweep, is helpful to think with that, although it is too simplistic of a kind of transition.
But I think what you're doing there is making a really important point, because what ties all of this together, I think, is the shift in a way is not perceptible to everyone, from sort of a top-down kind of eugenics to a bottom-up kind of eugenics. And the bottom-up eugenics has both positive and negative components, right? In terms of, the positive aspects are all of these catalogs you can look at if you want to, pick a sperm donor, and what are the criteria that, or the traits that you think are attractive. And, it's not a huge spectrum that's going to be in those catalogs for people to look at. One of the things that is interesting in that context is the way in which assumptions about race and ethnicity play out. There was a court case in the United States not that long ago, brought by a number of women of Asian descent, who argued that there was price fixing happening in terms of how much they could be paid for their eggs that they wanted to sell. And they were saying there's a unique market because people want Asian eggs, right? Because most of the eggs that were available were from white women, college students mostly, and of a very particular type and model set of assumptions about what are the desirable traits. So what's interesting is that I think we've all gotten to a point where we understand it's not great when the government makes those kinds of decisions to either, encourage certain people to reproduce and to impede the reproduction of others, both your positive and negative eugenics. But when we think it's bottom up, we think individual people are making private reproductive choices, that's fine. And I think what we need to understand is those private choices are made in a socialized context where people bring to bear a number of problematic understandings about what is or is not desirable, and also a failure to appreciate that at some level, some of that's fashion, and it will change. And change perhaps within the lifetime of these new beings that you're creating, having made certain choices about what is or is not a desirable trait. So I think that's a really important point that you're bringing up, Professor Stern, where you're actually, saying it's just the modern techno-eugenics that we're living through now. And we need to see it for what it is, and not to sort of think, well, that's okay, because this is individual people that are empowered and making individual choices. And I think you need to think about it in terms of agency and really what kinds of freedom is there to make what kinds of choices when you're, situated in a particular social context.
Okay, I'm going to start on a slightly different tack here. Another question from the audience. I do sociological research on CRISPR and see scientists dismiss concerns about eugenics regularly, despite attempts to include disability justice perspectives in public debates. I want to invite the panelists to imagine how might stakeholders mobilize outside of highly institutionalized decision-making settings? What kinds of social movements might we draw on as examples?
So I'll jump in very quickly. I've published a book called Altered Inheritance, CRISPR and the Ethics of Human Genome Editing. And in that book, I argue for two things that are thought to be controversial. One is I argued for a moratorium, a time-limited moratorium. And part of the reason for that was to say, we need to have time to have a conversation and to understand what it is that this technology can and can't do for us, and whether or not it's technology that we want to use, and if so, in pursuit of what goal. So that's the first thing I've argued for. And it's controversial because not everybody agrees that we need to have a moratorium. And some of those are scientists who are quite excited about the prospect of pursuing this science. But the other thing I've been arguing for is that whatever gets decided requires broad societal consensus. And that very much is a commitment to the view that the decisions before us now are too important to be left to the scientific community and to policymakers, that it is very important for the general public to have a say. And I talk about that in terms of obligations to education, engagement, and ultimately empowerment. So I think that that's really important. And I think this kind of a conversation is part of that process.
I think I would only add to these comments by highlighting the siloing that happens within academia. In training, geneticists or people in the sciences, they're not often asked to engage with people in the humanities and to think about their science in broader fashions. And I see this reflected in my own classroom when I have pre-med students versus the straight anthropology major. One of my challenges is being able to get these students to engage meaningfully, to think about how other fields might be useful in thinking about how you practice science. So I think that this is an issue that goes beyond sort of not only social movements, but in the ways in which we educate scientists in training.
I mean, as a historian, one of the things that I would point out is that we are engaging in a lot of hubris when we assume that the past is actually past. For example, that eugenics is something that chapter is closed. And, one of that chapter, so kind of thinking about how do these continuities continue into the present. And something that, I think would be good to look into is what has happened over the past 10 years in several states that have passed compensation bills to compensate really, with $20,000, $25,000. It's not a huge amount of money, but it does have symbolic and material value certainly to people for those who were forcibly sterilized by the state. This has happened in North Carolina, in Virginia, and potentially in California if the bill actually passes this year. And looking at those bills and those legislative processes really make us reckon with this past and see how it continues to affect individuals to this day who have been harmed, who are carrying those histories those histories of stolen reproduction in their very bodies today. So that is something that I think is important to note. And as our project at the Sterilization and Social Justice Lab, we've been fairly involved with the ELSI community, ethical, legal, and social implications of the Human Genome Project. And I would say at this point, things have really shifted over the past 10 years, and there's a lot of interesting work going on among some of the interdisciplinary researchers there who are taking a much more decidedly and confidently a social justice and a racial justice perspective. That is what we do in our work, and it's been appreciated. And so I'd be happy to share some of my kind of knowledge and contacts with the person who asked the question after the talk, if that would be helpful to them.
So I think the next question follows kind of directly on the heels of what you said. So Dr. Stern mentioned the destruction of records related to past state sterilization programs. That's obviously a major obstacle to historical research, but at least there are records in a number of states. Can you, Dr. Stern, and probably also the other panelists too, say something about issues of confidentiality and privacy with this kind of information, and then expand that to the kinds of things that Dr. Ben Torres and Dr. Baylis also spoke about. So this age of big genetic research where there's a lot of information out there.
Yeah, I mean, I think that's a very interesting question. In the case of Oregon, records were shredded. There was a flurry of activity in the early 20th century when the governor issued an apology for the sterilization law. And because the sterilization program went until the 1980s, you had in the late 1990s and the early 2000s, a good number of mental health justice advocates who were pushing for an apology and pushing for recognition of what had happened in the state. And there were some newspaper exposés that were written about the history. Some historians worked on it. And in that context, there was as I have learned about it, and if someone knows more, I'd love to be corrected. I don't know if some of these records have since been found, but a good number of the records were shredded by the state as a way of, trying to erase that history and to sanitize that history. That is obviously deeply problematic for a range of reasons. From the perspective of a historian, it's really a huge loss because we will not be able to reconstruct for Oregon the types of demographic patterns and capture the individual experiences that we're able to do for most of the other states that we look at. In terms of what the records, how are these records classified and how does that relate to confidentiality? In most cases, records relating to, and I'm talking about sterilization in institutions for the most part in the 20th century, most of those records are classified as, sensitive medical data that would fall under HIPAA protections. So in most of what we do in our research, we have to de-identify that. And if we want to tell someone's story, unless that story has appeared in a public forum, for example, it was published in the newspaper, or there was a lawsuit involving a particular person, unless that's the case, we use pseudonyms or abbreviations. In the state of California, there's a situation where there's a 75-year rule where records that are 75 years and older are actually completely public access. All of it, we could just upload everything we have to the web right now. We're not going to do that because that's ethically problematic and also would require a huge amount of work, which we're not prepared to do. But anything that is younger than 75 years, so that would put us, if we're in 2021, if I can do the math, of 1946. So our records dating from 1946 to 1952 are completely HIPAA protected. And I would say whether they're HIPAA protected or not, whether they're open access or not, it really still is an important question of whose history is this to share? How do you tell these stories in a way that both humanizes people whose reproductive autonomy was stolen from them and doesn't re-traumatize communities. And that's something that we talk a lot about in the context of the lab in terms of the ethics and the responsibility of sharing these stories. But for the most part, these records, like for example, the records in Iowa that we're working with, they can't even be photocopied. Everything has to be transcribed from you have to get the IRB and you have to get the release and permission from the state archives. But then, all of the records can be transcribed, and in the transcription, the idea would be you would not include someone's actual name, you might include an initial, because the rationale is to protect their identity under HIPAA, et cetera. So it's a really big question in terms of both working with the records and how are we doing it ethically, but also sometimes those ethical strictures, if we could call them, militate against humanizing the story and giving people who were subjected to eugenics the agency that they've already lost in the past.
The only comment I would make is that in a modern day context, we actually have often paperwork and tissue. And ultimately, the tissue is never going to be able to be completely anonymized. And that's a context in which I think the issues around privacy, again, are not always well understood. And I think Professor Ben Torres made the point, which I really want to emphasize, that we think of that information as somehow personal information, but it is actually familial information. And we haven't yet got our head wrapped around the idea of who actually needs to consent. And this could be in the context of looking for health information. Am I at risk of a particular disease? But once I find out information about myself, I've also found out information about, for example, my siblings, and maybe they did or did not want that information. I made the point also then with respect to paternity testing or other kinds of ways in which data can provide information, but you need to understand the information very often is not really personal. So even finding out about non-paternity has implications for the biological parent who you now may decide to find. You present yourself and they have no interest in having you knocking at their door. It can be disruptive within a family situation because for good or ill, a couple made a decision not to share that information. Maybe you feel violated that you had the right to information about your genetic heritage or not. So it's just to say that I think information is very complicated. But one of the things we're still grappling with is the difference between some kinds of information that can be destroyed and information lost. Whereas with the biological tissues, ultimately, there will be no real anonymity. You can de-identify for certain kinds of purposes, but if you need it out or want it, it's unique, identifiable information.
Yep, if I could just add on to that point, I'd actually like to share my screen. I have something from the New York Times that was published 2 years ago, but it drives home this point that our DNA is not just ours. So this was an article published, as I mentioned, 2 years ago. And basically, this is actually about a study that was published in Science. And what they say in this article at this point in 2018 is based on the genetic information that people upload to these third-party sites. At this point, it was 60% of white Americans could be identified, whether they tested or not, okay? The scientists that actually did this work, they did some back of the envelope calculations, and they said as soon as you have about 2% of the US population uploading their data to these third-party sites, 90% of white Americans can be identified. And again, it really just focuses on white Americans because that's who's buying and purchasing these tests. Another point that's really interesting to think about is this idea of anonymity with genetics and how it might be used. There's another company that I've been actually watching for years, Parabon Genetics, and they are offering services to various law enforcement offices in which they will look at DNA, they'll analyze it, and then they'll come up with a phenotype. So this is, for example, the picture they came up with based on this person's DNA. And you could say that the images look pretty similar. You could actually look through this and see some of the success cases in this, but this seems like something out of like Minority Report or science fiction, but it is here. And this company has been around for a number of years. So this idea, this illusion that we have of anonymity, of privacy, it's essentially gone when we start thinking about our DNA and where people are uploading it to.
So we've talked about a lot of things that have potential unintended consequences, that people participating in them, whether purposefully or not on purpose, may not have understood at the time or may not understand now. And some of these things can have bad consequences under certain circumstances. So I want to throw out another question to the panel and say, based on the kinds of things that we have talked about, what gives you hope for the future? Do you see a way that these unintended consequences might be pushed in a more positive direction, or there might be more education to push things in a more positive direction? What's your take on that?
I'll just start off by saying I think that recent awareness of the deep structural inequalities in US society and the long and unabated and ugly history of white supremacy is giving rise to new engagements with thinking about eugenics and thinking about the negative ramifications of some of these technologies. And I'll use another Oregon example, which I forgot to mention before. Some of you may know that in 1927, the US Supreme Court upheld Virginia's sterilization law in an infamous case, the Buck v. Bell case, in which Supreme Court Justice, Lead Justice Oliver Wendell Holmes Jr. said 3 generations of imbeciles are enough. And after that law was upheld, we saw a pretty much of a sustained and rapid increase going into the Depression years of sterilizations carried out in the United States until the 1960s and 70s. Well, that law has never been taken off the books. It's never been overthrown by the US Supreme Court. It's never been directly addressed by the US Congress or Senate. However, your senator, Merkley in Oregon, is actually interested in figuring out a way to tackle this law and to address it and to acknowledge its negative impact on US society over many years. So that actually gives me hope that I have spoken to a staffer from Merkley's office about the possibility of doing this, thought about what it would entail, and that this would bring visibility to this long history, show its ongoing relevance. So that's one thing that brings me hope. And I would also say conversations like this also bring me hope, and the fact that we can be speaking to each other across disciplines, which I think is really important, because our disciplines have different types of toolkits, and we can, in science studies more broadly, bring the elements or components of these toolkits, not to be too mechanistic about it, but these different frameworks together, and can hopefully make also our research accessible to wider audiences. So I guess I'm strangely hopeful. I'm usually not about this, but this panel has given me some hope, and I do see potentially good things going forward.
I think the one of the things that I am very hopeful about is I think we're having important conversations about difference and disability. And some of that has been spearheaded by the deaf community, pushing back against assumptions that have been made by people in terms of how certain genetic technologies should be used and what kinds of choices should be made. And many of that has been around different assisted reproductive technologies, where a deaf couple is actually seeking to have a deaf child. And people are saying, well, that's child abuse, that's terrible, that's crazy. And the deaf community having the ability to articulate, this is just about difference. We have a language, we have a rich culture, et cetera. And what makes your perspective on the world the right perspective? And I think that's also come to the fore with the most recent of the technologies, which is heritable human genome editing. Some of you may know that we do have 3 infants now born in China, where they were born of human embryos that had been genetically modified. And after that work was done, another scientist in Russia indicated that he was planning to do the same kind of research, also looking to modify genetic embryos in order to provide resistance to HIV. So that was the research goal. He was unable to identify people who wanted to participate in his research, and he was very cavalier about it and sort of said, yeah, well, you know, I've got some other projects, and that the 3 he named were dwarfism, blindness, and deafness. And again, there was a response that said, well, you know, who are you to decide that those are the things that can or should be targeted? And I think one of the things for me that's really important is we're not very good as a species, I would almost say, at just saying we're different. We always bring a valuative component, better or worse, right? Not just different. And I'm really hopeful that we can get to a point where we can actually genuinely embrace diversity and difference without having to make that kind of evaluative component part of that. And at least in this context, it's very often pushing to want to put certain things in the category of disability when they don't clearly belong there.
I would say the things that make me hopeful are really just echoing what Dr. Stern and Baylis have said. I think conversations like this that are cross-disciplinary give me a lot of hope. I am in love with genetics. I'm amazed with what we've learned and what we can learn. But I am also, I also recognize that it's a tool. And like any other tool, it can be used to build, it can be used to break. So I am hopeful that with continued conversations, more holistic ways of looking at the world and what diversity actually means, what difference actually means, I'm hopeful that we'll use these technologies, these tools in ways that represent the world that we actually really want to live in.
So we only have a few minutes left, and I'm going to throw out a final question. So I feel like this panel has been extremely educational. And of course, it's been open to anybody that can connect to it over the internet. But maybe we need to think about more general ways of educating or informing various groups of people about the potential use of genetics, whether that potential use is in a positive direction or a negative direction, but to think about, genetic determinism and how that might differentiate from cultural stories, but to make people have that more available in a general sense so they can understand it. And I wonder if you each could quickly say something about some way that you think that that might be accomplished.
I would say one way that might be accomplished really just involves community engagement, not only in research, if this is what you do, but also engaging in the community that you live in, which if that means finding ways to, talk to school kids, grade school kids about some of these tools and technologies, not necessarily that you have to get into the scientific mechanisms and details of all of this, but you can have a conversation with kids about, hey, we have the power to change things, should we? And get them thinking critically about these complex topics. So I'll be very brief because I know we're running out of time, but I really do think that there's an importance for people of all backgrounds to be able to have conversations together.
I think I would certainly echo that perspective, and I think I would say it in perhaps even starker terms. Those of us who have the privilege to work in the academy have a moral obligation to share our information. And I still think there are too many of my colleagues who think that their obligation ends, on the campus where they're employed, and that their obligations are to the people who have paid to sit in their classrooms. I really don't believe that. I believe that we have a moral obligation to share whatever information we have with the general public. And that means it behooves us to find creative ways to do that. The comment was made that all tools can be used for good or ill, and you can think about social media as a very positive opportunity to share information with a vast number of people that you don't have to know, you don't have to interact with, but you can take the time to share information. You can take the time to write, op-eds. You can take the time to write blogs. These things are not rewarded in the current academic system that we live in. And so I understand it's very hard, especially for junior scholars who are trying to meet the metrics that the academic setting has put out. But many of you will know that those of us in academia have a tremendous privilege, both in terms of the kind of work and creativity that we get to enjoy, but also tenure. And that comes with responsibilities. It comes with responsibilities to stand up and be counted. And right now, I think it comes with responsibilities to share information with the public. And if that requires more work than you will get credit for, so be it. I think it's really important. And I don't think enough of us do it. And at the very least, what we need to do then is train the next generation. We need to make sure that they understand their responsibilities more broadly and help give them the tools to do that kind of translational work and to reach out of the silos that can happen within that setting.
I'll just say that one of the things that I found in engaging with community activists and advocates around genetic and racial justice is that, many of the often our peer-reviewed publications are firewalled. People can't get access to them because they don't have an Elsevier subscription. They're not at an institution that does. So we are making efforts with our publications at the lab to actually make them public and open access so that we can just send the link and anyone can read it. And related to that, and I didn't have time to mention this in my presentation, but at our lab, we're also building, and I'm doing this with my colleague at Dartmouth, Jackie Wernimont, we're building a digital archive that we're calling Eugenic Rubicon, which focuses on the experiences and stories of sterilization in the US in the 20th century, with a focus on 3 of the states I mentioned, and bringing resources into that, and the idea of this is it's really geared towards a kind of a K through 12, maybe more of an 8 through 12 audience. Could be used in high schools, could be used certainly in undergraduate and community college settings, and by anyone else who's interested in it. And we have done focus groups for this project with folks in the disability rights community, with high school and middle school teachers. So really trying to get that feedback so that the stories and the histories that we're sharing both is an act, we hope, of some restorative justice, and also is accessible to a wider range of people. So that's one thing we're building, and COVID definitely has slowed us down. We hope to have a soft launch of this by the summer, but it's going to be about another 9 months. But I certainly will send the link to Professor Herman and anyone else who's interested if you would like to use that or delve into it at some point in the future.
So we're drawing to a close. I want to thank all of our panelists, Dr. Benn Torres, Dr. Stern, and Dr. Baylis, for all of their very provocative and insightful comments and conversation. I want to thank all of our audience participants. Unfortunately, we didn't get to all of your questions, many of which were very interesting and insightful also. And I'm going to turn this back to Professor Herman.
Thank you so much. I know that all of you out there will join me in echoing Professor Eisen's thank you to our panelists and to each of you for attending. We have a number of other public events coming up on similar topics or related topics. So if you don't know about them, please check out our website. I hope you found this 2 hours as interesting as I did. These issues relating to identity, belonging, ancestry, family are both personal, and it's obvious from the conversation, deeply social, with implications for our politics, our laws. you know, our ways of thinking and being, as well as implications for the way we do science going forward. And all of our panelists have given us so much to think about. Thank you so much on behalf of the Wayne Moore Center for Law and Politics at the University of Oregon.
Thank you.